Can you hear me? Yeah, I think you're muted. We can't hear you. Yeah.
Oh, I was muted. Sorry. Yeah, okay. Is my voice clear now?
Yeah. So we'll wait a minute. If any of the students wants to join, and then we'll start. Yeah. Actually, we have got only one question in the discussion forum. Also in the response sheet, I couldn't find any questions. So if the joint students have any questions, please post in the live chat box so that we can address the questions.
Amrita, can you paste that question in the chat box, please?
Yes. This is actually directed to one of the formulas in the population estimation. I'll just paste that so that everybody can see the question. This is related to the part 2 video of estimation abundance, where you discuss the n is equal to capital N is equal to mn by r.
Right.
So that's
two sample capture, what's called the canonical estimator. So this is something called the Lincoln-Peterson model. I'll just type that in the chat box. Model. There's an E missing in Peterson, but yeah, it's Lincoln-Peterson model. So the way it works is you have a population of unknown size. Right? You want to estimate what the size of that population is. You don't know the size of the population. So what you do is that you draw randomly from the population. Let's say these are birds, right? And you catch those birds using a mist net, and then you mark them uniquely. So every individual gets a unique, say, ring with a ring number. So it can be identified individuals. You can identify individuals. Then when you mark some proportion of animals in the population and you put them back in the population, the assumption is that they'll go mix with all the other individuals in the population which are unmarked, right? Now you have a population that has some proportion of it marked and some proportion of it unmarked. Typically, when working with birds and doing marking capture, a small proportion is marked and the larger proportion is unmarked, is not captured. So the idea is that if these marked birds go and mix in the population with the unmarked birds, with the next sample that you take, right, the proportion of birds that are marked in that sample, right, because they're mixed with the unmarked birds, the proportion of birds that are marked in that second sample should be the same as the number of birds marked divided by total population size. So the recaptured birds in the second sample, the recaptured birds divided by the size of the second sample should be equal to the number of birds captured in the first place divided by the total population size. And that's how you get n equal to mn by r. Capital M equal to lowercase m into lowercase n divided by lowercase r. So number of individuals captured in the first instance, number of individuals captured in the second instance is in the numerator, the number of recaptures in the second instance is the denominator, assuming that the marked individuals in the first term have mixed well with the unmarked individuals in the total population.
Sorry, hi, this was my question, and I just want to ask another clarification. When you're saying that it should be equal to the mark divided by the whole total population, like how are we getting to that should is my question.
But how are we assuming that it will be mixed?
Yeah.
It's an assumption of the Lincoln-Peterson model. In many cases, we can't assume. that the unmarked individual, the marked individual would gone and mixed well with the unmarked individual. We can't assume that. Because many birds are territorial, so you mark birds in one location, you're more likely to get that bird back than an unmarked individual in some other territory. Uh, so it only works in cases where this assumption is met. If this assumption is violated and there's some heterogeneity in captures, right? You're capturing birds and then putting them back in the population, but they're not mixing with the rest of the unmarked birds. Then you can't use this technique to estimate population size. There are more sophisticated models that do this in the closed model family of capture market capture. And in the closed model family, you can account for what's known as trap shyness. So when a bird gets stuck in a net once and then is released and it knows where the net is, it's going to avoid that net. So the probability of it being recaptured is lower than the probability of it being captured. You can account for things like that. You can account for heterogeneity in capture property, making males are more active because the female is sitting on the nest. And so the males have a higher probability of capture and the females have a lower probability of capture. You can account for that in those kinds of models. So in practice, although the Lincoln-Peterson model is sort of the canonical estimator, it's sort of like the most fundamental estimator under those conditions. The closed models are built upon that to actually be able to estimate these things better. In fact, the two sample market capture estimates are actually tend not to be very good. The more you sample, let's say you sample instead of just 2 days, you sample 6 days, and you are able to make the assumption that the population is closed within those 6 days, then that gives you a much better estimate of population size than the Lincoln-Peterson estimate.
Okay, thank you.
Sakshita, I hope that clarifies your question. Yeah?
Yeah, thank you.
Cool. So if anybody have any question, please post on the live chat box. If you guys don't have any questions, then we'll have to wind up in the next 5-10 minutes. Please make sure to use this session. This is for your, to clarifying the doubts before the exams. I can see 14 participants. Nobody have any doubts?
Actually, I don't have any doubt as such. I've heard the lecture more than once and could appreciate the importance or significance of most of the exercise. That's where it is. I'm trying to figure out and learn, and I'm enjoying. Yes, it's very well made talk. I should thank Professor Srinivasan. It is very well made. it looks a bit fast, but concepts are there, not going that fast. That way, still one can feel it is well made. Okay.
I see.
It's
You
very clear.
able to follow the lectures, right?
Yes. The clarity is there, very clear.
Yeah.
The language is very flowing a bit fast, but it is very clear. I can very easily grab that. Of course, I cannot make notes. So I put it in pass, go back to that, whatever it is, that transcript, make a few notes and
Yeah,
come back and run
you can
the
always
video. Because it's online, I can always pass it and keep going back and forth. So yeah, half an hour talk takes me almost about 2 hours to just go through one round. It's very nice, very nice, very
Thanks,
well made.
Gazama, for sharing your
Yeah.
thoughts. I think
Yeah,
you can download
illustrations
all
are
the
very nicely
transcripts.
made. Illustrations are very nicely made, and as long as one puts the effort, I think that things are there in place to appreciate and learn. Yeah.
Thank you
Thank
so
you.
much.
Thank you for the feedback.
Yeah.
Yeah. I think Smita, we can yeah.
I have a couple of questions in the chat box. Smita asks, I'm studying effects of blasting and crushers and effect on birds, but unable to find proper data. As in, Smita, you want to collect data on this, or you want to find data that already exists? Now, you want to collect data, so you want to design a study that looks at the effects of blasting and crusher. So this blasting and crusher, what is this really? Is it like stone quarrying and mining kind of situation? I see. Okay. So I think basically what you would probably have to do is find wherever the quarrying or mining is happening, whatever habitat it was earlier, natural habitat it was earlier, you'll have to find um, um, in that undisturbed habitat of that kind, then have multiple replicates within that habitat for sampling birds. So let's say you're sampling the bird community using point counts. Then you want to have multiple point counts in the location without the blasting and the crushing, and then a similar number of point counts in the location with blasting and crushing. And then sample birds in those locations, and then we compare what happens to bird populations of bird communities because of blasting and crushing. I think there will be more information regarding study design and setting up how to design a scientific study to ask and answer a particular question in Suhail's version. I think there is one Suhail's lecture. There is one lecture on study design in ornithology. And has that lecture happened yet? It's happened, no? It's
Finished. The
yeah.
lecture design is already finished, but you always can post this question in the discussion forum so that he can reply there. Or I'll send the question to him. Like, if you have any specific questions on which research design methods that have already covered.
Yeah, I think it's difficult to give very tailored, precise advice regarding this because I don't know what the landscape configuration is. I don't know where that quarry is, what the original habitat was. I don't know what the bird community in natural habitat is expected to be, what the bird community in the quarry is expected to be, and the number of replicates are made, and the number of samples are need might be different in the natural habitat versus the quarry, because there are fewer birds and fewer species in the quarry, and so therefore you need less effort to be able to characterize the community. All these considerations that go into study design. So if you can give me a little more details later, I could potentially give you some more specific advice. But I think a good place to start would be going with its lecture on quantitative ecology and designing system. Another question, what are the standard procedures for collecting avian saliva of fluid samples? Furthermore, while fecal analysis helps determine bird's diet, what specific information does saliva provide that other samples do not? Right. So the standard way to collect saliva is with a cotton bud, just roll it in the beak of the bird. What you get from the saliva is not actually components of the saliva itself. What you're getting are epithelial cells from the cheek, and the epithelial cells contain DNA. And so that's one way of getting the DNA of the bird from the epithelial cells that line the mouth inside the bird. For blood samples, it's exactly like you would do for us when we get tested at the doctors and we have to give a sample from the brachial vein, which is the vein in front of the elbow. It's the same brachial vein in birds as well, and it's the same basic technique for collecting the blood samples as you collect blood samples for humans. Fecal analysis, yeah. So saliva provides actually epithelial cells for DNA rather than any dietary information. But a much better source of DNA is blood in birds, because birds have red blood cells that have nuclei, and the nuclei have DNA. Unlike us, where our red blood cells don't have nuclei, they have a nucleus. And so our red blood cells don't contain DNA, whereas the bird nucleus, bird red blood cells, because they have the nucleus, they contain DNA. And so DNA is used in a variety of ways. If you collect a DNA sample, if you collect a blood sample and you extract the DNA from it and you look at the sequence, DNA is composed of 4 what are called bases, A, T, C, and G, and they're arranged in, if you look at the sequence and the way in which those 4 bases are arranged, then you can estimate, for example, relatedness between 2 species, what time they actually split, how many million years ago they had a common ancestor, and then they split into 2 different species. You can even do very detailed population genetic analysis to look at relatedness between individuals and ask. So you have a set of islands, and there's birds moving between the islands. Which island is contributing birds to which other islands based on the DNA patterns, based on the DNA sequence patterns that you see. So it's a very it has many applications. Can we use non-invasive environmental sampling, collecting droppings from the ground to study their diet, or does the risk of contamination make it necessary to handle the bird directly? I am probably not the best person to ask this question. So yes, there will be contamination if the fecal sample is on the ground, and the risk of contamination is high. And especially when you have an environment where you can have multiple sources of DNA, you might be picking up DNA from organisms that the bird is not actually eating, but is there in the environment, and then you might wrongly conclude that the diet of the bird contains that particular species or whatever as well. The other disadvantage of bird droppings out left that have fallen on the ground is that the DNA degrades pretty fast. So all bird droppings are not going to be as useful as fresh bird droppings in terms of getting DNA from the droppings. So as the sample is exposed to sunlight and so on, the DNA degrades. And so if you have a fresh sample and you collect that fresh sample from the bird itself and you store it immediately in a buffer or any kind of fluid that preserves the DNA, then that's a much better source of information regarding the diet of the bird. So from watching live bird cairns, I've noticed that chicks seem to wake up or become active just because before their parents actually arrive. How does the bird community communicate this? Do the chicks have a way of sensing their parents' arrival before they are even visible? I'm sorry, I have, I have no idea about this at all. But yes, I think you're right in that just before the parent arrives, the chicks wake up and start open their beaks and start begging. Um, Perhaps behavioral, Margari might be better able to answer this question. Um, it might just be, they hear the approaching parents with the wing beats, and so even before the bird lands, they know that the parents are coming. But that's just a hypothesis. I don't know the answer to this question. But, if you could post that on the forum, then maybe Manjari would be able to answer that. But if there are any questions on populations and communities, would be glad to answer those. And the techniques for studying populations.
So do you guys have any questions related to bird population and communities estimation, population estimation? Please post the questions. We'll wait 5 more minutes.
You're welcome.
I have some experience to share, if you, with your permission.
Yes. Sure,
Okay.
sure.
In Tamil Nadu, there was a raptor statewide raptor assessment program undertaken. It was a 2-day program. 1 day was a orientation program on 31st of January, and 1st February was the program. They were saying that this is the first raptor assessment taking place in the country. What they had gone through some systematics. Now that I went through the lecture by Dr. Srinivaswa, I could appreciate it, the significance of that a lot more. I participated as a volunteer, and there were forest department staff, and we were there. Some of us were there as this thing. What they had done is the whole state they had made into grids of 32 kilometer by 32 kilometer, the whole entire state. And then for each district, they had taken certain 10% of the grids. So I happened to be in Madurai. In Madurai, there were about 9 grids were selected, which constitutes the 10% of the entire area. And they have taken care not to repeat neighboring grids, and they have made that. And it was a 1-day program, and they were the actual counting started. Counting was done in 2 modes. One was either with the vehicle. The people who go on a vehicle is supposed to cover about 20 kilometers in about 4 hours time. It's more than 4 hours. Starting from 8 o'clock to 12 or a little bit on minimum that much. And after going every 2 kilometers, they were supposed to stay at least minimum 10 to 15 minutes, take data, and move to the next point. And there was the same grid. There was people going on foot. The on-foot team is supposed to go about minimum of 6 kilometers, and again making the counts and recordings. And they were how to make the whole data. There was a data sheet provided. And also one interesting thing what I saw was they had given a mapping, what they call as a map locus was a software which they had given. That software, once it takes, each of these grids were given grid numbers. So, and the grid number is put, and the entire that topography is taken. And as we are walking with GPS, it keeps marking the entire track. And also it has the facilitation wherever we take a photograph or a video or make some comments, all these options are available. We can go to that software and select. If we are clicking a photograph, we click the photo, we say I'm making a photo, and click that, automatically at that point that was clicked with all that location, it just takes like that. And that way it is done. And in Madurai, we had 9 groups going. And raptors, it was not that many in numbers. And still in the group what I went, I was one volunteer. There were 4 forest staff, forest officials or forest officers. And we cover, I went joined the foot group, and we could make that count. It was a very nice exercise. And all that data they had from that software itself. Once the data is accumulating, when we have to submit, we have to take that, and just from there we have to export the data. So that way they authentically whatever has been done systematically, it's basically sampling. And that data is transferred with all the details, whatever comments, whatever photographs, whatever videos we are able to take. And periodically, we have to also go to some places, take some photographs, and keep submitting it. It is centrally all are gathered, and then a report is formed.
So you have learned the sampling method and how you choose the, in that sampling method, you did, I think, transects, right? Where you walked.
We were walking and making point count.
Yeah,
Point
that is transect method.
Location. We were not having any gadgets used, as it was suggested, compass and just that distance.
Okay. Did you do the like after the survey? Did you do any measurements? Did you calculated the the abundance from your walk? Did you do any sessions?
We didn't do any of. We submitted all this data to the forest department. They would have done whatever.
Nice. But you had your personal data with you. Maybe you can go through the analysis on your own, just to learn the things.
This raptors thing, there were not too many. Okay. In our whole say foot this thing survey, we could see only 4 instances in the whole 4 hours. Okay, 1 sikra was seen. Okay, 2 black kites were seen, because they were so few in numbers in the area what we were covering. So that way they were minimal. And they also conducted a wetland bird census, and another one territorial bird census they had conducted. I was part of that. That were there in reasonable numbers. Okay.
I see.
This territorial bird census was conducted on February 16th or so. And in our group, we could get about 52 birds recorded. In reasonable, some of there in some numbers also. Yeah.
Yeah. Thanks, Gautama, for sharing. Do you have any doubts when you did the survey related to this lecture? Like how the sampling size, it's an important thing to understand, right? What, after the survey, how did you choose the survey technique? No.
It's basically counting. Okay. Counting.
I understand it's a bird count, rapid count.
Point count, basically.
Point count.
Point count.
Okay. So I think in Umesh's video, you can find out, figure out how you calculate this bird abundance from these surveys, right?
Yes, I think
that. I have to tell that, Gautama, it's, raptors are quite, it's quite difficult to quantify raptor population sizes, because they're so mobile and they move very large distances. That's probably the reason why neighboring grids were not surveyed. And also the reason why it was all standardized at the same time, so that the chances of double counting become low. In that if everybody's out at the same time surveying different grids, then you're not likely to count the same raptor in two different grids. Anyway, the chances of doing that are minimized. If it's simultaneous and with the neighboring grids excluded. Standard point counts, if you read papers, that, for example, estimate bird population sizes in forest birds, for example. You'll often find that there'll be acceptance of the methods that will say, yeah, we use point counts, we replace standard, whatever, we stood for 10 minutes and we point counted all the birds, and blah, blah, blah. And then at the end of it, I'll almost always do a sentence saying, raptors, swifts, and other birds that flew over the plane were excluded from the analysis. And that's just because you don't know where they're going, what they're doing. Whereas with the birds that are in the vegetation, they tend to be a little more sedentary in terms of territory use. Whereas the swifts and the raptors and so on tend to move quite large distances.
Yeah, see, that to some extent that is taken care of in that what we had done, because only 10% of the area is being covered. The entire grid, they had made about, for Madurai, it was about 180 grids, in which we are just going to only 9 grids. And if there is, it's the adjacent ones are as far as possible avoided. So that way, even there is some movement, probably that is a little bit taken care of to some extent, of course. Yeah. And
now, ideally, in an ideal world, right? If you want perfect information, you have to survey all those grids, or every single one of those grids, right? And that's what's called a census, where you're not taking a sample of the habitat, but you're counting every bird. That's a census. But this our logistical and manpower constraints mean that censuses are pretty much more or less not possible in most cases, and so therefore we need to take a sample of the habitat, a sample that is representative of the habitat that is being sampled, and then extrapolate from that sample. So if you're saying 10% of the habitat was census and it had 20 birds, then you want to be able to make a reasonable claim that the entire habitat has 200, roughly 200 birds. Because if you extrapolate from that 10% and the number of birds on that 10%, then the entire habitat should have that many individuals. So that's the approach that's used in sampling.
Yeah. Okay. Yes.
I have another question from Ashta. A very sensitive question about the bougainvillea chitla. What is the biggest threat to the current population of the bougainvillea chitla? Why is it a challenge to increase their numbers and their habitat? That's something we are currently researching, right? As we speak, this research is in process. We don't know. The species was first cited by Professor Ramana in 1995 and formally described to science only in 2006. So we don't know the history of this population. We don't know whether the bougainvillea chitla has always been rare, whether it was much more common in the past and has become rare now. What is the minimum viable population size that's required to support the species? All of this we are working on right now. With this, from the genetics of the bougainvillea chitla, we can tell what the population size was in the past, when did it crash, and try to correlate that with other climatic events like ice ages or changes in the habitat that we know from the past. Changes in habitat happen in these particular times. Whether that's what's responsible for the small population size. The threats right now to the species are inherently one, the fact that it has a small population size, and two, that it is geographically highly restricted. So the total population size is between 14 and 20 individuals, and the total geographic range size is about 2 square kilometers. Now that's what makes this species vulnerable to 2 kinds of randomness, which is technically called stochasticity. There's demographic stochasticity and environmental stochasticity. Demographic stochasticity means randomly in some year, let's say a majority of the nest failed, and most of the chicks died in the nest. Then that can bring the population down to below minimum viable population, causing it to go extinct. That's what demographic stochasticity is. Environmental stochasticity is especially applicable when populations are restricted in space. So if you have a bird species with a large range size, then one extreme event, a flood, a drought, extreme snowfall, which is localized to one area, is not going to have a big effect on the entire population of that species. But one major snowfall, um, extreme event, one major snowstorm in that 2 square kilometer area of the species can cause it to go extinct, and that's what's called environmental stochasticity. The other risks for this species are, of course, climate change that might cause the species to shift its range up and then go extinct because it reaches the tops of mountain and has no more space to go to. Right? Um, uh, While the challenge to increase the numbers in their habitat, right now, currently, what we think is this. Um, and this is just a hypothesis. We're working on this. We're trying to figure out if this is the case. The bone leucicula, like its closest relative, which is called the Asian leucicula. I'll just put that in the chat first. The Asian leucicula, which is found in southern China. Both of them are species that don't use primary undisturbed forests. They need some level of disturbance in the habitat. So they're what are called early successional species. So early succession is a stage in forest regeneration where the forest has not yet become mature, or what we call old growth forest. So because because the Bougainvillea tritium we think is early successful and needs some amount of disturbance to maintain that kind of habitat, and that kind of habitat is becoming rarer and rarer in the state because earlier what would happen would be that would be shifting cultivation process by which you burn down one patch of forest and grow crops on it for 3 or 4 years, but the soil is depleted and you move on to another patch of forest. And then you have this patchwork of different plots that have been farmed in the past of different ages. So some might have been left 50 years ago, some might have been left 3 years ago, some might have been stopped cultivating on some of those 20 years ago. So you have a patchwork of forests that is regenerating. And so you have multiple types of habitat. Now what's happening is, I think that could have been good for the Bougainvillea, what we think, because we always have these young patches of forest that have been recently abandoned after being cultivated. What's happening now is that shifted cultivation is no longer practiced in any significant form or any significant scale in the area of the Bougainvillea habitat. It's either forest that is old growth or permanent agriculture. Now that it's, once you clear that land is planted every year, and it's very pesticide and fertilizer intensive, and so that's what allows that permanent agriculture to happen. So you're not getting what we think is we're not getting that intermediately disturbed status habitat anymore, because shifted from that shifting cultivation kind of landscape use to a more settled, permanent settlement, permanent agriculture kind of use. That's what we think. You're welcome. by the observation on the Bougainvillea club. It split from the emission in Bougainvillea club started becoming two different species about 1 million years ago. It's a very recent split. They're very, very closely related. So 1 million years ago is not early at all. It's a very recent split between the emission and the Bougainvillea club. And so we think that, and by the way, the emission huge class pretty well studied. There are captive populations and there have been field studies of the emission huge as well. And we think that based on the fact that there are recently diverged, not a very old split, the Bougainvillea should have very, very, very similar behavior and characteristics to the emission club. So we have sort of better information from emission in terms of population parameters and so on to try and model what might happen to the Bougainvillea population in the future using population models.
Thanks, Umesh, for that wonderful explanation. I think, yeah, this was a detailed study. Could you please share the paper you are referring where these two splits happening, like for two population emerged, emission?
It's never been published. We just found out.
Okay.
We just did the lab work ourselves in
Okay, cool. So if no questions, then we'll wind up.
अस्सलाम वालेकुम
Yeah, sure.
मैडम मैं बेसिकली पाकिस्तान से हूँ और मैं पीएचडी स्कॉलर हूँ इधर यूनिवर्सिटी ऑफ काड़ा में। तो मैंने आपका कोर्स देखा था लिंक्डइन से, तो मैंने इसको ज्वाइन किया। लेकिन मैम जो आपकी टाइमिंग थी जो कोर्स की, मैं इसमें मोस्ट ऑफ जो क्लासेस हैं वो नहीं ले पाया। जो ऑनलाइन मीटिंग थी मैं नहीं अटेंड कर पाया क्योंकि मैं ऑलरेडी लेक्चरर हूँ यूनिवर्सिटी ऑफ लाहौर में। तो मैं मूव कर रहा था तो ड्यूरिंग जो है वाली मेरी ट्रैवलिंग जो है आपकी क्लासेस होती थी, तो मैं नहीं अटेंड कर पाया। तो अगर वो पहले शुरू में हमें जो है ऑनलाइन आपके लेक्चर मिल गए थे, लेकिन अब वो भी नहीं मिल रहा है। तो काइंडली अगर कोई पॉसिबिलिटी है, कोई लिंक आप शेयर कर दें जो हमारे मिस हैं लेक्चर्स, हम उनको अटेंड कर सके।
So this is regarding the recorded video lectures, right?
बिल्कुल बिल्कुल बिल्कुल।
So all the lectures are available in the NPTEL portal. Even these live session videos are available in the live session tab. So in the dash menu, you have to select the live sessions. Under that, all the recorded videos are available.
ओके ओके मैम, तो मैम इसका कोई लिंक वगैरह शेयर कर देंगे आप?
So it is available in the NPTEL portal where you are seeing all the weekly lectures. So along with that, all the weekly sessions are available. So I can do one thing. I'll attach the links for all the weekly sessions. Yeah, in the discussion forum. Sure.
Please,
I'll do that. Yeah. What are the specific adaptations?
एंड मैम थोड़ी मुझे ये भी इंफॉर्मेशन चाहिए थी कि आपका जो ये एक सोसाइटी है, सेपरेट सोसाइटी है या गवर्नमेंट के साथ कोई लिंक आपकी सोसाइटी है?
This society, I didn't get the question. So
madam,
yeah,
I want to say that this organization attached with government or independent organization.
NPTEL, right? Yeah, this is an association basically.
Okay.
Where we arrange classes from IITs, ISOs. So we have faculties collaborating for delivering these classes.
Okay.
Yeah.
Because I am also
this is an effort from IIT
association in Pakistan, Wildlife and Fisheries Care Association. So I am looking for forward collaborations and this type of collaborations in which we share our experiences and data regarding ornithology.
Yeah, so this is an organization like collaboration work, but for delivering lectures on ornithology.
Yes, yes.
Yes.
Because in Pakistan, specific in 2018 to 22, I am working on a bird, Bia weaver.
Why we were.
जी. Because in Pakistan most of breast and जो मतलब केनाल रिवर्स वगैरह के ऊपर जो कीकर के दरख्त थे वो मोस्टली जो है वो काट दिए गए। तो फिर जो है वो इसकी जो नेस्टिंग थी वो एक सिंबल एक हमारे पास दरख्त है उसके ऊपर शिफ्टिंग हुई सिंबल के ऊपर। तो उसमें जो है इसकी जो नेस्ट था और जो एग लॉस था वो बहुत ज्यादा था। बिकॉज उस प्लांट के ऊपर जैसे ही हवा का कोई झोंका आता था तो नेस्ट जो है वो गिर जाता था। मेरे पास उसका वीडियो रिकॉर्ड और वीडियो डाटा काफी था। लेकिन हमारे पास वो क्योंकि जो कीकर का दरख्त था वो नहर के या जो दरिया वगैरह उसके बिल्कुल ऊपर होता था तो वो एक स्ट्रॉन्ग जो है कह लें उसके ऊपर नेस्ट की फॉर्मेशन होती थी। तो वो नेस्ट की फॉर्मेशन स्ट्रॉन्ग ना होने की वजह से काफी सारा जो है ना वो लॉस हुआ था। तो मैं चाह रहा था कि मतलब इस तरह से कोई कंबाइन जो है वो हम आपस में डाटा शेयरिंग भी हो और इस तरह से जो क्लासेस वगैरह हैं कोई हम पाकिस्तान के प्रोफेसर भी और आप लोग भी मिलके हम आपस में चला जो है वो मिलके
Yeah, this is an
क्वेश्चन करें।
online course where faculties will answer to your questions based on the lectures they are delivered. So if you have any specific questions, you can always post it on the discussion forum. If you are not able to actively participate in these live sessions, please post the question on the live session. Yeah, based on the lectures they have delivered. Yeah, thank you, Mom.
Thank you.
So I think one more question. What are the specific Yeah.
Chatbots from Gavilia. So what are the specific adaptations for these two speciation in reference to Google specific adaptations for these two specifically? So the way that the speciation usually happens is there's one single population that gets split by some kind of barrier, geographical barrier that prevents gene flow. So it could be that the bone Leochicla and the Amazon Leochicla's ancestral species had a range that included the Himalayas as well as southern China. And then something happened about 1 million years ago that caused the population in China to become separate from the population in the Himalayas. And so there's maybe some mountain range came up at that point and split the two populations. And then the two populations then diverged on separate trajectories. One becoming the Bougainville and the other becoming the Amazon.
Hello, am I audible?
Yes.
Yeah, so I just wanted to ask, your voice was breaking. So that part when you explained that due to the gene flow, there was a breakup from the same population. After that, I lost you for some time. There was an echo and I couldn't clearly hear you. Could you please repeat that part?
Yeah, no, I was just basically saying the ancestral species that gave rise to both of these species, the Bougainville and Amazon probably had a range that included China as well as the Himalayas. And then something happened in time that separated these two populations. Maybe a mountain range came up, or a deep river gorge, river changed its course and separated these two populations. And after the populations were separated, they were unable to exchange genes because they're not interfering with each other. And so genetically they start diverging and they start becoming different different species.
You are talking about genetic drift, right?
Part of it could be genetic drift. Part of it could be even selection based on the locations, because even at the same location over time, you don't have the same environmental conditions, right? You'll have ice ages, and then the ice ages will retreat, and then where you had ice age, forest will come back, and so you can also have selective processes operating there, where given that the Western Himalayas the Eastern Himalayas are different from the Himalayan mountains, maybe you can have selection of different types of selection on the Bougainville and a different type of selection on the Himalayan to adapt to local conditions. So it could be both, could be selection. Definitely dispersion happening because there's no gene flow between these two populations. And the other process that can change genetics is mutation. Most mutations tend to be harmful and not good. And so, mutation, I don't know, but mutation probably played a relatively smaller role than drift and speciation in causing these two species to diverge.
Right, Dapalina, is that answer your question? Was that audible? Okay, yes. So do anybody have any questions? We have 5 more minutes. Okay, I think we'll wind up the session. It's almost time. Thank you, Sumesh. Thank you, Umesh, for joining. Thank you so much.
Thank you.
Thank you, everyone, for coming up.
Thanks,
we'll
everyone.
meet.
Thanks for joining. And thank you, Amrita.
Thank you.
Thank you.
So we'll join for the next week, weekly live session with Dr. Suhail Gadar. Yeah, you will get the announcement regarding the date. Thank you so much. Thank you, Umesh.
Thank you.